Hey,
Okay, we'll just give people a couple more minutes to trickle in. We'll get going in another minute. All right, we'll get started. For those wondering, the picture behind us is of Summer Lake and Winter Ridge. This is in Oregon. Well, welcome to the first in a series of winter webinars hosted by Western Watersheds Project. My name is Adam Bronstein. I'm the Oregon Director here at WWP. We're excited to have all of you here with us today. After the presentation, we'll try to get all of your questions answered. There's a Q&A box down at the bottom of your screen, so you can enter in your questions. And again, we'll try to get to all of them. Roger Long is an accomplished ecologist with a focus on nitrogen cycling in the sagebrush-steppe ecosystem. His research emphasizes the critical role of nutrient dynamics in ecosystem health, with a special interest in sage-grouse recovery. Following a 20-year career in corporate information systems, Roger returned to academia to earn advanced degrees in biology and ecology. His professional journey spans teaching, research, and conservation advocacy. So today's presentation is titled How to Starve an Entire Ecosystem. And we'll learn how livestock grazing and agriculture disrupt nutrient dynamics in the sagebrush steppe with a focus on nutrient flows, nitrogen flows. Roger will shed light on how these disruptions impact plant and wildlife communities, offering insights into the challenges and opportunities for ecosystem restoration. So over to you, Roger. Thanks again for joining us today. It's good to see you.
thanks, Adam. So yes, my presentation is on nitrogen cycling, anthropogenic effects on nitrogen cycling in the shrub steppe. And there's good news and bad news. The good news is that agriculture has a positive effect on nitrogen availability within a limited area around the agriculture, and that grazing, as you might guess, has a negative effect on nitrogen availability. And so we'll go ahead and show how that works. So I think we're all aware that the sagebrush habitat is in trouble. It's continually being damaged or destroyed. The picture on the left is fairly typical of what you would see in southwest Idaho these days. That is shrub steppe. You can see the remains of a sagebrush plant there in the foreground. It's been destroyed. It's gone. What's been left with is a cheatgrass desert. And this is almost certainly due to fire. The destruction is pretty much complete. On the right-hand side, what you see is sagebrush steppe that is probably more typical of what you would see these days. It looks like healthy sagebrush habitat, but if you really get out into it, you'll notice that there's almost no plant diversity out there. This particular site, there are probably 5 species of plants. There's sagebrush, rabbitbrush, quested wheatgrass, cheatgrass, and a lupin, which is toxic to cows, and so they leave it alone. And that's what you'll find there. So there's the destruction of habitat that we can easily see. That's on the left there, fire, roads, fragmentation. But almost as bad, at least for wildlife, is the destruction that we can easily see. And in many cases, it seems to be from a loss of nutrients. There's a change in species composition, and there's a loss of diversity involved. So my dream was to be a community ecologist, but I just kept falling into systems ecology. It seemed to go to the roots of most of the problems that I was examining. And so basically, it's the study of nutrients, how they enter ecosystems, which we call influxes, how they cycle through the ecosystems, and how they leave the ecosystem, which we call effluxes. So the idea is to elucidate the root causes of productivity at landscape ecosystem scales. So that was the level at which my project was conducted. And so the big 5 nutrients that we are interested in, most interested in, are carbon, of course, we are carbon-based life forms, oxygen, hydrogen, nitrogen, and phosphorus. So carbon, oxygen, and hydrogen are generally pretty available. You know, they're either there or they're not, especially in the case of oxygen and hydrogen. Most of that comes into the system through rainfall or through the atmosphere. Nitrogen is interesting. There's some limitations to nitrogen when I'm going to get into in a little bit here. And phosphorus, and we just don't know much about phosphorus because it's just in the ground and it's there or it's not there. So what my study is based on is something called ecological stoichiometry. And if you guys have ever done any college level chemistry, you're familiar with stoichiometry. It's the method of balancing chemical reactions. And it applies at ecosystem scales. So it's involved in determining the proper ratios of carbon, nitrogen, and phosphorus in the ecosystems. If the ratios of those nutrients are out of balance relative to the life forms in that ecosystem, then the nutrient becomes what we call a limiting nutrient. And usually on terrestrial ecosystems, nitrogen is the limiting nutrient. Phosphorus is a limiting nutrient in marine ecosystems. So back when I was teaching at College of Idaho, I did this very same presentation actually. And I started talking about nitrogen as a limiting nutrient. And one of the students, definitely not one of my ecology students, kind of sat back and looked at me and says, nitrogen is 78% of the Earth's atmosphere, and just kind of sat back and looked at me like you just blown my whole argument out of the water. But it's the unique chemistry of nitrogen that makes it problematic. Atmospheric nitrogen occurs as N2 bonded together with a triple bond. Okay, so that's the most powerful chemical bond, and that makes the atmospheric nitrogen very difficult for plants to utilize. Every chemical reaction involving atmospheric nitrogen is going to be chemically uphill. So plants can only obtain that atmospheric nitrogen. Well, they don't obtain atmospheric nitrogen. Actually, they partner with symbionts like Rhizobia, Frankia, and macrobiotic crust to change it into forms they can use, like nitrates and ammonium. And they use those to build their tissues, build proteins and DNA. So the limitations on productivity in the shrub steppe, we know it's not a very productive environment. It's cold and it's arid, and that slows down all sorts of biological reactions that take place, like decomposition, nitrification, even photosynthesis. So because it's obviously arid, it's usually thought of as being strictly limited by precipitation. They're not really looking at other nutrients as being potential problems. So my hypothesis was that the habitat decline could be a result of anthropogenic nitrogen loss, an efflux. And that is, well, what would cause that? And so the response I came up with was probably grazing. But to examine that, we need to understand a little bit more about how nitrogen exists in the ecosystem and what forms it can take that are usable and forms that are not usable. So an ecosystem ecologist will basically divide the nutrient supply in an ecosystem into various pools. So I look at the soils, the soil pool of nitrogen, the plant pool of nitrogen, the animal pool of nitrogen, and the atmospheric pool. And what I'm really interested in here is what's in the plants. What's in the soils is not accessible to animals at all. In fact, some of it is not even accessible to plants. And we know that the atmospheric nitrogen is not accessible either. So what I centered my study on was what the nitrogen content of the various plants were in a grazed ecosystem and an ungrazed ecosystem to see where that nitrogen was going, what was happening to it. So why do I pick on cattle? Okay, because every herbivore alters the environment by grazing, trampling, defecation, urination, all of that stuff. And they all graze selectively. They all have preferred plants that they eat. And so why would cattle be any different? How do cattle affect the ecosystem differently than all the other native animals in the ecosystem? And the difference is they cause a loss of nitrogen because they open the nutrient cycle. And they cause nitrogen to leave the system. And that nitrogen leaves the system in the form of cattle biomass. In other words, so when cattle gain weight by grazing on the shrub steppe, those nutrients that it acquires from grazing leave the system when they leave the system and go to a slaughterhouse, and they end up being consumed in someplace far, far away from where the nutrients came from. And those nutrients do not make their way back. So livestock are about 3% nitrogen by live weight. So for every 100 pounds of cattle biomass gained by grazing on the shrub steppe, about 3 pounds of that is nitrogen. And when you think of all the millions and billions of cattle that have grazed on the shrub steppe over the last 150 years and been exported to other places, you can see this is a fairly massive effect. So what my hypothesis was that nutrient export has a detectable effect on nitrogen dynamics in the shrub steppe. If it's not a detectable effect, it doesn't mean anything. It doesn't affect the ecosystem. So the other effect, the other anthropogenic effect, is that sagebrush herbivores feed in agricultural fields. Every farmer out there will tell you that everything from rodents to elk will come into their fields and consume their crops. And those agricultural fields make poor cover, so generally after the herbivores have gained enough nutrients from the agriculture, they leave and they use the nearby shrub steppe for cover. They eliminate wastes, and therefore by doing that, they transfer nitrogen from the agriculture to the shrub steppe. So this nutrient transfer should also have a detectable effect. This is my other hypothesis. And this was what I originally designed my experiment around, and then actually the grazing hypothesis came in later. So I used stable isotopes in this study because they could give me clues as to what was happening with the nitrogen in the system. And the reason for that is that plants preferentially take up the light isotope, the delta 14N, and we call that process discrimination, okay? When they are limited, when the plants are limited by the available nitrogen, they also begin taking up the heavy isotope, which is delta 15N. So so if you are comparing plant tissues, the nitrogen content in plant tissues, and you see a higher percentage of delta 15 in one plant than another plant, you can safely assume that the higher delta 15N is due to nitrogen limitation. In other words, there's some reason that the plant has to utilize the less preferred form of nitrogen. Actually, any difference from atmospheric nitrogen, which is delta 15N equals 0, any difference from that shows some discrimination. So the carbon-nitrogen ratio is the ratio of carbon to nitrogen, obviously. A ratio of 25:1 or below indicates a nitrogen-rich plant. These plants are going to be the ones that the herbivores are going to graze on preferentially, okay? Grasses actually tend to have a carbon-nitrogen ratio of 40 or above. So the reality is that any herbivore out in the shrub steppe is going to preferentially eat plants other than grass. Grass will be a last resort because the carbon-nitrogen ratio is the lowest. However, I will explain later in the presentation why cattle can subsist on a pure grass diet more easily than native herbivores. So I made a series of predictions, okay, on the differences between the grazed site and the ungrazed site. There would be less discrimination during plant uptake on the grazed site due to lower availability of nitrogen. So what we would see with the plant samples from the grazed site would be a higher CN ratio. Now, this didn't exactly come to pass, and I'll explain what actually did happen. So longer-lived plants with deeper root systems will show higher delta 15N and fewer differences between sites because they are essentially taking their nitrogen from deeper soil horizons. And those deeper soil horizons have higher delta 15N because there are denitrification and deamonification processes that go on that result in nitrogen volatilizing from and basically breaking down ammonium and nitrates breaking down and leaving a residue of higher delta 15N. And older plants, for many of the same reasons, also have delta 15N. So shorter lived plants, and this is critical, with shallower root systems like grasses and forbs, will have less delta 15N and show greater differences between sites. Lower nitrogen availability will also result in higher carbon nitrogen in plant samples from the grazed site. So the experiment was designed around being able to measure different distances from the agriculture and what effect distance from the agriculture had on nitrogen availability. And so in terms of wildlife biology, this is not as interesting as the overall grazing effect, but at least it does explain why you see a lot more wildlife around an irrigated alfalfa field. Not only is the alfalfa nice and green and tasty, but it has much higher nitrogen content than the surrounding plants because it's a legume. It's a nitrogen fixer. So again, the prediction was that there would be lower carbon nitrogen ratios in plant samples from near the agricultural fields. So these were my study sites. So the grazed area was down here near the town of Howe. There's the INL is very selective about where they will allow cattle to graze on the National Laboratory. And since there is no open water on the INL, they can dictate where the cattle can graze simply by only permitting water tanks in those areas where they want the cattle to graze. So this Birch Creek site, because of archaeological resources, the INL didn't want cattle in there, and so they denied the BLM permission to put water tanks in there. So this area has not been grazed in probably 100 years. So now it's interesting. So occasionally you can see the Big Lost River here. Most years it dries up above the highway here. And this is what they call the Lost River sinks. It just, somewhere up here, it just starts sinking into the ground. Occasionally they'll have an extremely wet year, maybe 1 in 20 years, where the river actually reaches the INL, but that's not typical. So these are my study areas, and these are my numbered quadrats. Okay, so what I had 55 quadrats, 5 by 5 quadrats in each of these sites. These sites were 2 kilometers by 2 kilometers out from the agriculture and 3 kilometers across. And that's a lot of sampling to do. So we broke it down into 5 by 5 quadrats. And then we took plant samples from 3 transects across each 5 by 5 quadrat. So that actually gave us a pretty good picture of a 6 square kilometer area without having to sample the whole 6 kilometer area. So I divided the study area into 10 intervals of 100 meters each, and they started at the agricultural border. And then we put an 11th interval about 1,000 meters beyond the 10th interval, just to see if there was any major difference between 1,000 meters from the agriculture and 2,000 meters from the agriculture. And if we did, that would mean we would have to do more sampling. Unfortunately, we did not find that. So we randomly placed 5 sampling sites within each 100-meter interval, and then we put the 3 transects across each site, and then we harvested all of the above ground plants that actually touched a transect. If it was a shrub, what we did was we harvested 25 liters of the new growth from each shrub. So we had 5 species that we sampled for the stable isotopes. And the reason we only had 5 was there were only 5 species of plants that were found in sufficient abundance on both sites to actually make those comparisons, to have valid analysis of variance comparisons. So we had Indian ricegrass was pretty common. Of course, big sagebrush was common at both sites. There was a bottlebrush grass that was common at both sites, gray rabbit brush, and winter fat. And winter fat was the highest nitrogen content of any of the plants that we sampled. And that really surprised me. But then again, that's probably why winter fat is not common in the shrub steppe anymore. It used to be, it's not now. I was really surprised that there was very little cheatgrass in my samples. But cheatgrass apparently does not do as well at the higher elevations. We were between 5,000 and 6,000 feet, our sample sites, whereas Southwest Idaho, which has just become almost entirely a cheatgrass desert, is more like between 2,000 and 3,000 feet elevation. So basically, what this is going to tell you is I found significant differences. There isn't any, and some of the differences were extremely extreme in that between the delta 15N and the carbon nitrogen ratios. There was a difference in carbon nitrogen ratio, both with site and distance. But the interesting thing to me, as part of this, I studied the book, Ecological Stoichiometry. And what ecological stoichiometry predicted was that the same species of plant between the two sites would have different levels of delta 15N and different carbon nitrogen ratios. What I found was that within each species of plant, those ratios were pretty much the same. What was different between the sites were the plant species that I found. I'm going to skip that. So to get to the meat of this, plants on the grazed site have higher 15N, indicating possible limitation. Plants further from the agriculture also have higher delta 15N and higher carbon nitrogen ratios. So we're saying that livestock grazing has a negative effect. Also, agriculture exhibits a positive effect on nitrogen availability. So herbivore optimization is a hypothesis that came from an African ecologist named McNaughton. He observed herbivores in savanna in Africa, and his findings were that low to intermediate grazing increase above ground plant productivity. But the thing about that is those are actually higher rainfall areas, 30 to 40 inches a year, that are maintained as grassland by elephants essentially bulldozing all the trees. So they use the BLM and other grazing entities use this hypothesis to justify high levels of grazing in semi-arid environments. So we found no indication of that. So the vegetation cover and biomass we predicted would be higher on the ungrazed area. And so we predicted that we would find no evidence of herbivore optimization. We also predicted that species richness would be greater on the grazed side. That sounds a little anti-intuitive, but other studies have found that areas that are prone to disturbance, like grazing, will actually have higher numbers of species, but they will have less species richness, which depends on species evenness. In other words, so the grazed area was dominated by those 5 species of plants I talked about earlier. But there were many examples of only one species in one individual of a given species in all of our quadrats on the grazed side. In other words, you had a lot of rare species, whereas on the ungrazed site, there was very little evidence of that. So to treat our samples, this is methodology. The only thing is, if people are concerned about sample sizes, we took 4,000 plant samples, and we treated all of them the same way, analyzed them for carbon nitrogen ratio and delta 15N. So species richness was greater on the grazed side, 32 to 27 species, whereas the diversity indexes were greater on the ungrazed site, and that's the result of fewer rare species. The interesting thing, there were a number of forbid grass species on the grazed area that we only collected on one sampling site. In other words, they were rare. So cover on the ungrazed area was 1.37 times that of the grazed area, highly significant. Biomass was almost double. And so we used cover simply because the correlation between cover and biomass was extremely high, and there was no need to analyze anything but cover. Our statistical analysis showed that the differences in cover were mainly between sites, really highly significant differences between the grazed site and the ungrazed site. So I looked at, there were several species in the analysis that showed up with extremely low carbon-nitrogen ratios. These were, in other words, these plants had extremely high nitrogen content, and surprisingly, cacti was one of them. And they were so rare on the grazed site that I couldn't do a classical ANOVA. I had to use this non-parametric test called Bernadette-Monk analysis. And what it showed was that these three plants were almost entirely confined to the ungrazed site. I found almost no MBC stands for macrobiotic crust. I found almost no macrobiotic crust on the grazed site, and I found very little cacti and bryophytes. And that bryophyte is extremely high in nitrogen. It's 18%, and that's almost the nitrogen percentage of meat. So, and the significance of the macrobiotic crust is that it's been shown that that is the major contributor of fixed nitrogen in the shrub steppe ecosystem. We have very few legumes, very few other sources of nitrogen fixation in the shrub steppe ecosystem. So what does it all mean? So the delta 15N was lower on the ungrazed area. So my hypothesis was that the lower delta 15N indicated greater nitrogen availability, which in turn should predict that you're going to have more cover and more biomass. No great effect of distance from agriculture for cover or biomass, even though there was an effect for carbon nitrogen. And again, that effect was simply that the species composition was different. Higher nitrogen containing systems species were found only on the ungrazed site. So lower carbon nitrogen adjacent to the agriculture make agricultural areas even more attractive to herbivores. So the lower carbon nitrogen ratio equals higher quality forage. And so the plant nitrogen pool, going back to our nutrient pools, the mean of carbon nitrogen for all plants was slightly lower on the ungrazed site. However, the biomass was almost double. And the total nitrogen in plant samples on the ungrazed site is approximately double. And that's not even factoring in the really high nitrogen-rich plants like that moss, which is called Tortula ruralis. It's interesting. So if you're out there in the shrub steppe and you see a black mass around the root, around the base of rabbit brush or sagebrush, and you dump a little water on it, it'll instantly turn bright green, and that'll tell you it's that Tortula ruralis. So the interesting outcome of this experiment was by doing this plant sampling, you can almost draw up a sort of a back of the envelope nitrogen budget for the entire plot. You can extrapolate from those quadrats how much nitrogen is in each system. So there were some questions originally early on in the study of where the nitrogen on the grazed area was going. People were reluctant to admit that the cattle might be the problem. They brought up effluxes of nitrogen because of volatilization. However, that should be the same on both sites. Wind and runoff, the wind essentially came from the same direction on both sites, should affect both sites similarly. And there were no influxes to the ungrazed area that are not also present on the grazed area, except macrobiotic crust. And the lack of that macrobiotic crust is almost certainly due to grazing. And that's not just me saying it, that's Ehrlich's 1993 study pretty much ascertained that. So this is our nitrogen pool in the shrub steppe, and you can see some mule deer happily feeding in an alfalfa field, jackrabbits, sagebrush, sage grouse. And so the importance of the nitrogen to native animals in the system versus cattle is explained by something called the Kleiber curve. I don't know how many of you people are familiar with that, but it was originally constructed as to illustrate a relationship between size and life expectancy. I mean, I think we all know that the bigger an animal is, the longer its life expectancy is. The interesting thing is over time, we found that it applies to metabolism and it applies to tolerance for toxins. So the metabolic rate of most organisms scales not 1-to-1, but to the 3/4 power of body mass. And what that means is that as animals get larger, as you're looking at larger and larger animals, their metabolic rate will be lower for each gram of body mass. Okay, so in other words, large organisms have more efficient metabolisms. They can subsist on forage. The other thing is that ungulates also have symbionts that can convert cellulose and lignin to digestible sugars, and that gives them a big advantage on feeding on lower quality plants because those consist mostly of cellulose and lignin. Small herbivores lack those symbionts. They have a less efficient metabolism. They need more nitrogen in their food. They also have smaller digestive systems. So plants with a low carbon nitrogen ratio are their preferred forage. They're preferred forage for everything, actually. But for smaller herbivores, they are necessary, okay? For cattle, they prefer them, but they can do without them. So the cattle on an ecosystem on the shrub steppe system, can eat all the high quality forage and then continue to subsist on the poor quality forage like crested wheatgrass and essentially starve out the smaller herbivores. So this is not a part of the animal pool of nitrogen in the sagebrush steppe. This animal is an efflux. It is not going to die on the shrub steppe. Its nutrients are not going to be contributed to the shrub steppe when it dies. Those will be removed from the ecosystem and consumed somewhere else. I didn't deliberately choose this photo because of the cheatgrass all around the cow, but come to think of it, it's kind of appropriate. So my conclusions were that livestock grazing negatively affects the nitrogen budget for plant cover, biomass, and species richness. It also affects forage quality by changing species composition. So the high nitrogen containing plants are not found in heavily grazed areas. So the agriculture positively affects forage quality, but not at great distances from the agricultural fields, and it doesn't affect the quantity. And that's probably due to native herbivores feeding in the agriculture and then taking cover in the sagebrush. So my approach was unique that I was able to elucidate fine scale patterns across the landscapes. Just in the interest of time, I did not include my IDW maps, but you can really see how patchy the nitrogen availability is, especially in the grazed area, if you have access to those maps. And I'm hoping, actually, if you request those maps from Adam, he has a copy of my dissertation that he can get those to you. So as you might expect, I confronted some arguments with the whole study. And the first one, most ranchers simply believe that basically you have, you feed the cows, the waste products come out, and it's a net zero. And, that's actually somewhat beneficial. And the issue is that the livestock biomass gained by feeding on the shrub steppe, it's when that biomass goes to slaughter, it becomes a net loss of nutrients. In other words, it's not the grazing itself that's the problem. It's the removal of the biomass gained from grazing that takes nutrients out of the ecosystem. So the other one, grazing is grazing, right? There should be no difference between animals and livestock. And the answer to that is, for the most part, nutrients from the native animals stay in the system. Nutrients from livestock do not. And the last argument was, well, those nutrients got to be replaced somehow. And nobody can tell me how, especially when you bring up the fact that the major source of nitrogen is the macrobiotic crust, and that is one of the first things to go when cattle are introduced to the ecosystem. So you talk to a farmer, and they can pretty much tell you by how much was harvested and removed from their farm what they need to replace with fertilizers. Ranchers don't seem to think that way. So anyway, the farmers understand that they have to replace nutrients. No nutrients are being replaced in grazed areas in the shrub steppe. So after 150 years of grazing, and I would hesitate to guess how many millions of tons of nitrogen has been removed from the system, we're really starting to see the effects over the last 30 years. For a long time, you didn't really see those effects. I mean, when I first started going out in the shrub steppe in the 70s and 80s, you could still pretty much see the same animals. I go out in the shrub steppe now, I don't even see jackrabbits, much less sage grouse. It's an event to see a jackrabbit around here. I live in Northeast Nevada, and they are just not very many. Adam, I'm winding up.
1 minute warning. We have got some questions.
Oh, okay. So what's the first question?
Well, go ahead. Why don't you give us a conclusion there? You got another minute.
That is the conclusion.
Okay, very good. So Laura Welp, our Southern Nevada director, is going to ask some questions, and I hand it over to Laura.
Thanks, Adam. Our first question is there any data on how buffalo affect these environments in comparison to cattle, and how they affect nitrogen levels?
Well, to do that comparison, you would have to go someplace like Yellowstone National Park and find an equivalent area outside the park where the buffalo aren't available. I think we can infer from the study, from the results of the study, that bison, again, it's not the species that's the problem. It's what happens to the animal after it's dead. Okay, if it dies in the ecosystem and it decomposes in the ecosystem, then the nutrients stay in the ecosystem. For the most part, during the great buffalo slaughter back in the 1870s, except for tongues and hides, most of the buffalo were left to rot where they died. Okay, so that was pretty much a net zero for the ecosystem. Whereas with cattle, they're almost universally transported out of the ecosystem to be consumed elsewhere and not decomposed in the system. That answer your question?
Don't know, but I'm going to keep on going.
Okay.
Here's a question about the biological soil crust. Alan Savory once claimed biological soil crust as a last life left situation due to removing herbivory from the system. What is your take on this?
I would not. What my experience was in the shrub steppe was it was about a first life removed. And it's never been scientifically verified, but I think it is a symbiont with certain types of plants in the ecosystem. And apparently those plants are among the first to go when cattle are introduced to the system, because where I saw grazing, I did not see macrobiotic crust.
Okay. What is the relationship between this and the massive loss of A and O soil horizons over the last 150 years?
Well, those are due to erosion. Okay, the massive, most of that is due to erosion. I did not see significant effects of erosion in my system, and I don't know that it would be a major source. I mean, most of this stuff is flat. I mean, just flat. The water just doesn't drain off. It just soaks right in. And there's not a lot of precip out in the shrub steppe, and most of what there is snow. So I think in other systems, which I know a lot less about than I do shrub steppe, it's mostly due to erosion. I know a little bit about the Palouse up in southeast Washington, and that, yes, they have suffered massive losses of topsoil due to erosion, wind erosion, water erosion, just, and it mostly from agriculture, really. It just, they've been taking the topsoils down over, the last 150 years. So, but in my study area, no, it didn't seem to be significant.
Okay, we have some questions on regenerative grazing. So you mainly discussed grazed versus ungrazed, mentioning the herbivore optimization hypothesis as a matter of animal density. In regenerative grazing, the number of animals is far less relevant than the duration of rest, the period without grazing animals that allow for recovery, and the impact of grazing. This way, regenerative grazing mimics seasonal migration patterns. In your research, have you looked at the timing of grazing and rest? This information would make your research extremely relevant for regenerative versus degenerative grazing.
Well, I'm not really all that familiar. I mean, I've read up some on regenerative grazing, and I think in other systems that are more productive, it's probably really valuable. The thing about the shrub steppe is simply that it is a low productivity ecosystem due to the fact that it's both arid and cold. And so regenerative processes are slow. This is the problem with the nutrient export, is that the system itself replaces those nutrients slowly. So you'd have to look at the timescales of regenerative grazing. And I did not look at that. I don't know what those timescales would be, but I would suspect that they are decades rather than months or years.
Okay. Let's see, I'm trying to. Someone asks, wouldn't it make more sense to feed the corn, soy, and alfalfa to humans directly?
I didn't do any research on that, but it makes sense. You know, feeding lower on the food chain. Um, you know, I mean, we can eat alfalfa, I guess. It's better as sprouts, though.
Okay. This somebody does a follow-up on their soil on that erosion question. Um, says nearly all of the sage steppe had. This ignores the fact that nearly all of the sage steppe had any significant herbivory prior to the European invasion.
Oh, it had tremendous herbivory. It was incredibly productive. The early settlers, you know, crossing northern Nevada, were talking about herds and herds of antelope and bighorn sheep and flocks, clouds of sage grouse and sharp-tailed grouse. The difference was that the nutrients those animals gained from grazing in the shrub steppe stayed in the shrub steppe. That is the difference. It's not the herbivory. It is where those nutrients go after they've been herbivorized. I think I just made up a new word that's never going to enter the vocabulary. Yeah, it again, it is not it's not the herbivory, it's not the grazing. It is the transport of nutrients out of the ecosystem as a result of that grazing.
Right, thank you. Are there data on historic migration of buffalo in this region? Indigenous people lured buffalo into these dry steppes with controlled burns, thus extending fertile areas. That's kind of outside your area of expertise, but
everything I have read, and admittedly it hasn't been that much, is that there were not the great herds of bison in the Great Basin that there were on the other side of the Rockies. They were here, but they were not incredibly abundant, and they were not dominant like they were east of the Rocky Mountain front on the Great Plains. The early settlers mentioned them, but they were not like, you know, great herds of buffalo as they crossed the plain. In fact, they talk more about pronghorn antelope and bighorn sheep.
okay. Someone asked a question about referring to the elephants you were talking about, the African herbivores, and wonders if there's domesticated or wild species in that
They
state.
were wild. They're wild. I mean, that's what essentially what maintains those grasslands over there. Those grasslands get enough rain where they should probably be forested, but the elephants have been, you know, elephants' metabolism is so efficient that it can eat trees. I mean, not just the leaves, you know, branches, bark, the whole thing, and digest it and live on it. In fact, in Southeast Asia, you know, they're with the elephant trainers and elephant keepers in Southeast Asia, different species of elephant, but when times get tough and they can't afford feed, they feed them cardboard. They feed the elephants cardboard, and they can live on it. They don't thrive on it, but they can live on it. So it's yeah, the ultimate effect of the Kleiber curve there. Yeah, they bulldoze the trees, they knock them over, and then they eat them.
It seems that relying on animal death to restore nitrogen would be highly selective as to the area affected. Do you see nitrogen islands where animal death occurs?
No, because animals are essentially, I think of them as a nutrient transport system. You know, if a cow dies in the shrub steppe, what's going to happen is it's going to be scavenged. And those animals that scavenge that, there probably will be some extra nitrogen deposition there, but the animals that scavenge that dead cow, vultures, coyotes, foxes, you name it, are going to spread those nutrients all over the ecosystem.
That's good. By consuming the beef locally and not shipping it out of the area, does the nitrogen then stay local?
Well, that depends on what the local community, how the local community treats their sewage. Okay, because that essentially is where the nutrients end up. I debated on my original presentation for my dissertation, actually showing a toilet to, having a slide of a toilet to show what actually happens to these nutrients. But that is the crux of it. It's, if it's consumed by humans, it's going to go into some type of, whether it's a very primitive sewage treatment, like an outhouse, or a very sophisticated, like some of the larger cities have, where they actually create biosolid fertilizer, those kinds of things. So it depends on what's done with it. If it's left to the animals, they figure it out. They do what needs to be done.
Could this degeneration be mediated by fertilization of the steppes?
Well, okay, this is a thing. It would have to be done on a massive scale. In fact, Adam and I were talking yesterday about a project where they're putting salmon carcass analogs in streams to replace the natural runs of Pacific salmon that have been eliminated. And they're hoping what they're hoping to do is bootstrap the stream ecosystem by putting these salmon carcass analogs. And I think what they're doing is they're taking spawned out fish from hatcheries, and they're sterilizing them, and then they're putting them in the streams. And they're showing a very positive effect of that. But a stream is a much more limited in area than, vast acreage of shrub steppe. And I don't even know how to calculate the amount of fertilization you would need. I think the expense would be staggering. It could be that distribution of some type of carcass analogs in specific places could bootstrap the nitrogen fixing processes in those places, and then that could spread through the ecosystem. But that's going to take time.
Still a lot of questions coming in, but let's do one or maybe two more.
Okay, I wish that grazing research would not be formulated as grazing versus no grazing, but rather look at contextually appropriate versus inappropriate grazing in that ecological context. Can you imagine how your research method around stable nitrogen isotopes could also reflect beneficial grazing impacts on regenerative farms?
Well, I mean, it could show you where the nitrogen is going to some extent, but again, the issue with grazing is not whether it's beneficial or detrimental. Okay, grazing is neutral. I mean, it's been going on in the shrub steppe since forever. It's what happens to those animals that are grazing on the system. Do they get to stay in the ecosystem? Do they die in the ecosystem, redistribute their nutrients through the ecosystem, or do they get transported off and go someplace else, and those nutrients disappear from the ecosystem? That's really the crux. To me anyway, at least that's what my research indicates.
All right, we're at 1 p.m. Mountain, so let's wrap it up. Roger, thanks for your time. That was a great presentation and some great questions. Sorry we didn't get to all of them, but we have future webinars coming up, so please stay tuned. If you support these events and the work of WWP, please consider making a donation. I just dropped a link in the chat. So thanks again to everybody for participating today, and hope you have a good rest of your day and into the weekend.